Okay, so in this video, we're going to talk about some principles of gene expression. So the gene products rise and fall in response to the molecular signals, and they regulate the gene expressions. So you have inducible genes and repressible genes. So inducible genes, the normal state is to not to be expressed, and then when they're induced, they're expressed. And repressible genes, they're on all the time, such as the housekeeping genes or the constitutive gene. So they keep on making them, and then they're repressed whenever they need, there's too much of it. So the constitutive gene expressions so stay more or less constant. So there's two levels of control. You have transcriptional control and translation control. And transcriptional control is more efficient because less energy is used, because you don't have to make the mRNA and then break down the mRNA. If you just stop the transcriptional part, then it's more efficient and more energy conservative. So we have a couple of different methods to analyze RNA. We have a ribosomal blot, no, I mean a northern blot. We have, so what it is separation of the RNA molecules on gel, and then you take a probe with a radioactive, you take a radioactive probe and incubate it with the gel, and then the gene of interest should be seen by a change in color or like fluorescence or something like that. And so here we have the cytoplasmic RNA and then the nuclear RNA. So we see all these different plots in the nuclear RNA because you have splicing going on inside the nucleus. So here this part is both exons and introns, and then you have some splicing happen, some of the intronic material is taken out, so it's smaller, it keeps on getting smaller, and this would be all exonic material. And in the cytoplasm, all you get is the exonic material since there's no splicing going on. And this is very tedious, takes very long because you only look at one mRNA at a time. And the PCR reaction is a little bit faster, and you can do 4 or 5 instead of just one at a time. But still, it takes pretty long. What's really fast is this DNA microarray, which you can do like all these different things at the same time, and then the color indicates whether the mRNA is found or not. And if there's a regulatory sequence, if there's a difference in the promoter sequence, that can account for a 1,000-fold difference in the mRNA product. The major determinant for expression of housekeeping genes, major determinant being the promoter sequence, and the deviations from the consensus sequence between the RNA polymerase and the promoter region would usually prevent or reduce transcription of the mRNA. And their specificity factors are like sigma factor in bacteria and the Tata box binding protein in eukaryotes. So the repressors, they impede the access of the RNA, access of RNA polymerase to the promoter. So this is negative regulation, and this is usually found in prokaryotes. And activators, they enhance the binding between the RNA polymerase and the promoter regions, and this is called positive regulation. Activator, positive regulation; repressor, negative regulation. And over here we have different sigma factors being activated based on the cell needs. So if the cell is being heat shocked, Then sigma 32 is activated where more chaperones are being made because of sigma 32. And then sigma 28 for genes are activated for the stationary and the stress response. And if the bacteria placed in nitrogen medium, nitrogen metabolism is needed, so sigma factor 54 is on, so the genes that are needed for the nitrogen metabolism are being activated. And here's a diagram of the repressor and activator. So if you add the ligand to the repressor protein, the conformation changes, the repressor comes off and the gene turns off. Or you can take off the ligand and then the conformation changes and then the gene turns off. So over here you're adding the ligand to get rid of the repressor. Here you're taking off the ligand to get rid of the repressor. And same thing basically for the activator. You have the activator here, you add the ligand and the activator comes off and the gene gets turned off. And then you take off the ligand and the ligand take off the ligand from the activator protein comes off and the gene turns off. Or if you want to think about it the other way around, you take off the ligand, the activator binds and the gene turns on, and then over here you add the ligand and the activator binds and the gene turns on. You take off the ligand, the activator binds and the gene's turned on. That's about it for this video.